multiple alleles. As per the Mendel law, every character is controlled by one pair of alleles. That is, there can be only two options, out of which one is dominant and other is recessive. With Sin's last portion of polygenic inheritance, it was proved that it can be more than two genes or alleles which exhibit the same characteristic of an individual or organism. And therefore, cases were observed where there are more than two optional forms of a gene for the same character. And so, multiple alleles on this basis can be defined as when three or more alleles are responsible for the same characteristic then such alleles are known as multiple alleles. Multiple alleles possesses the alleles that occupy the same specific locus on the chromosome. That is, they are present on the same specific locus of the chromosome. For example, let us study the concept of multiple alleles with the help of blood group of the humans. Example, the blood type in humans includes mainly three, which in combination forms blood group A, B, AB and O. So, out of this, these blood groups are formed on the basis of antigen and antibody that are present within the individual. Thus, A contains the antigen A and antibody small b. These antigens are present within the RBCs while antibody is present within the blood plasma. Similar is the case of another blood group, that is B, in which the antigen that is present is B and antibody small a. Similar, the blood group AB contains both antigens, that is antigen A and B, but lacks antibody, that is no antibodies occur within it. Similarly, the blood group O contains no antigen but contains both the antibodies. That is small a and small b. In this manner, within the blood group, various genes are responsible for producing the antigen and antibody. Thus, within blood group, the inheritance is based on the three alleles that is IAA, IB and small I, out of which IA and IB are dominant while small I is recessive. Therefore, this dominant allele responds to the antigen. Therefore, IA codes for antigen A, while IB codes for antigen B. Correspondingly, on the basis of presence of antigen, the blood group can also be decided, like antigen A represents the blood group A, while antigen B represents the blood group B. Similarly, the recessive allele, that is I, is considered as a non-producing gene and therefore it represents for the antibodies. In this manner, the blood group and the corresponding genotype can be obtained. Like the blood group A consists of genotype or can be possibly IA into IA or IA into small I. Second, the blood group B can have genotype IB or capital IB and small I. Similarly, the blood group AB can have genotype only IA and IB. And for the blood group O, it will contain only two small I. That is, it contains both the antibodies while AB contains only antigen. Therefore, in this manner, if the blood group of the parents is known, then the probable blood group in their children can also be known on the basis of presence of this genotype. Conversely, if the blood group of the child is known, then the blood group of its parents can also be known. This description of blood group was first proposed by the scientist named Landsteiner. He was the first one who described about human blood group. That is, he depicted or resulted out that the occurrence of four types of blood group, the presence of antigen and antibody within it. And so, he gave the result that there are four kinds of antigen that are present on RBC and which kind of antibody occurs in blood plasma within the person. This figure describes the reaction between antigen and antibody that takes place within a person and correspondingly the blood group that is formed. As it can be seen, this describes the antigen, this the antibody, and correspondingly the blood group is obtained. Such a reaction, that is compatibility of blood group, is generally checked out with the help of serum test. That is, serum test helps in understanding or checking the blood transfusion. Within a person. Moreover, sometimes within it clotting also takes place. Clotting of the blood occurs when the antibody reacts against the antigen and causes agglutination of RBC. Which possesses that antigen. That is, this RBC contains that antigen. Due to this agglutination, clotting of blood occurs. On the basis of antigen and antibody present within the blood group, it can also be decided that which blood group person can become a donor or recipient. So, a blood group person containing blood group A can donate to persons both containing blood group A and AB, while it can receive only from the person that possesses blood group A or O. Similarly, for another blood group, that is B, a person can donate to the blood group containing B or AB and receive from the person containing blood group B or O. Similar is for the AB, where it can donate to the person only containing AB blood group, while can receive from people of all the types of blood group, and therefore it is considered as a universal recipient blood group. This is because AB blood group contains both type of antigen, and therefore it can receive blood from any type of blood group. On the contrary, the person with O blood group can donate to any person containing any type of blood group, and so it is considered as a universal donor. Because it contains antibodies, both small a and small b, as a result it can donate to any person, but it can accept only from a person possessing blood group O. This figure depicts that in which type of blood group agglutination takes place. As it can be seen from the figure, the agglutination takes place only when the donor blood group contains the antigen that is exactly opposite to the recipient blood group. In this manner, clotting or agglutination of RBC occurs. And therefore, a person containing blood group A will agglutinate with the person containing blood group B. In the similar manner, donor blood group B will agglutinate with the recipient blood group A. And therefore, in this manner, agglutination takes place. This figure depicts the paternity style on the basis of blood group that is present within an individual. That is, for example, if the child contains blood group A and that of mother is O, then the father should contain blood group capital A or AB, because it cannot be B or O, because the A blood group corresponds to the A antigen, and therefore the blood group A or AB is essential. In this manner, the other blood group are arranged, which describes the paternity of an individual.